Can I know why you're late?
I didn't set my alarm yesterday. Sorry.
Are you ready for class today?
Yeah.
Are you sure? Are you excited for class today?
Huh?
Hello, class. How are we doing today? Hopefully everyone's well. Maybe next time we won't be too late. So today, as you can see written on the board, we will be looking into nanobiotechnology, specifically next generation sequencing and other DNA sequencing alternatives, with a focus on one technology, which today we're going to be looking into Oxford Nanopore Technology.
Imagine this. You're a detective working in Manhattan, New York, and you just received a phone call that there's a murdering in the NYU dorms. You enter the site as a detective, and you see that there's blood on the floor, and it spikes some curiosity. Now this blood has been confirmed that it's not the victim's blood. So how can we find out what the...
So as bioengineers, using the blood that we just found on the floor,
how can we reveal the identity of the killer? Yes?
We compare the color of the blood to everyone else's blood.
Comparing the blood? Oh, okay. Okay.
Professor, I think there's something called DNA sequencing we can do. I think that's the correct approach.
Exactly. Round of applause, guys.
So DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions essential for the growth, development, functioning, and reproduction of all living organisms. It's made up of two long strands that twist together to form the iconic double helix structure. The strands are connected by 4 types of chemical bases: adenine, thymine, cytosine, and guanine. These bases pair specifically A with T and C with G to form the rungs of DNA ladder. The sequence of these bases determines the genetic information that directs cells on how to produce proteins and perform various functions. This DNA sequence is what makes every living organism unique. In fact, every human has a unique DNA sequence, making it a powerful tool in fields like medicine, forensics, and genetic research. Here in Abu Dhabi, government initiatives are leveraging the power of DNA through groundbreaking projects like the Emirati Genome Project and the Population Genome Project. These initiatives, driven by government collaboration with the private sector, aim to map the genetic information of the Emirati population. The objectives are ambitious: to advance precision medicine, where treatments are tailored to an individual's genetic makeup, to identify genetic predispositions to diseases for early intervention, and to contribute to global genomic research. The scope of these projects is immense, considering the human genome contains approximately 3.2 billion base pairs in a single haploid set of chromosomes. Despite this complexity, the Human Genome Project, completed in 2003, laid the foundation for understanding the genome. This project was a global effort to map the entire human genome for the first time, which obviously is going to unlock new and better opportunities, whether for personalized medicine, disease prevention, etc.
Opportunities for personalized medicine and personalized treatment.
Equally as relevant is something that we call next generation sequencing. So the next generation sequencing is a set of DNA sequencing technologies that allow us to map the A's and T's and G's and C's to a genome, which we can then use, unlike Sanger sequencing, which used to be the traditional method. Next generation sequencing is a way for us to do it more rapidly and a lot more sequences at one time. So the way next generation sequencing differs exactly from Sanger sequencing is...
In Sanger sequencing, the process begins by taking a DNA sample and separating it into single strands. A short primer binds to one of these strands, providing a starting point for sequencing. Next, the DNA is placed in a reaction mix that includes regular nucleotides, the standard building blocks of DNA, and a small amount of special chain terminating nucleotides, called dideoxynucleotides or DDNTPs. Each DDNTP corresponds to one of the 4 DNA bases, A, T, C, or G, and is labeled with a unique fluorescent color. During DNA synthesis, the enzyme, which is in this case DNA polymerase, builds what we call a new strand by adding nucleotides. When a DDNTP is randomly incorporated, instead of a regular nucleotide, it stops the synthesis, creating a DNA fragment that ends at that specific base. Because this process is so random, fragments of varying lengths are produced, each ending with a fluorescently labeled DDNTP. These fragments are then separated by size using capillary electrophoresis. As they pass through a detector, the fluorescent labels reveal the sequence of bases, allowing the full DNA sequence to be reconstructed one base at a time, but however, from the shortest to the longest fragment. In next-generation sequencing, DNA sequencing is massively parallel, allowing millions of DNA fragments to be sequenced simultaneously. First, the DNA sample is fragmented into short pieces, each of which is prepared with special adapters on both ends so they can attach to a sequencing platform. These fragments are then amplified, creating clusters of identical DNA copies for each fragment. On many NGS platforms, sequencing occurs through a process that we call sequencing by synthesis, where fluorescently labeled nucleotides are added one by one to each cluster. When a nucleotide is incorporated, a camera captures the emitted fluorescence, which identifies the specific base, A, T, C, or G, that was added. This process continues with each nucleotide addition recorded for each fragment cluster, building up the sequence one base at a time. The result is a large volume of short DNA sequences that can be aligned and analyzed by computer software to reconstruct the original DNA sequence, offering greater efficiency and speed compared to Sanger sequencing.
So this high throughput sequencing technology has made it possible to sequence entire genomes very quickly, which opens up countless possibilities across various fields. So we'll now be looking into the uses of NGS DNA sequencing. And I just wanted to ask the class, does anyone have any ideas on what could be the possible uses of NGS DNA sequencing in their own medical fields or even in their own research?
From my respective research, NGS helps us to identify genetic mutations associated with diseases, enabling us to have more accurate diagnoses, and even allowing us for personalized treatment plans tailored to an individual's genetic profile. And with this technology, scientists can now sequence the DNA of cancer cells to understand how they differ from healthy cells, leading to the development of targeted cancer therapies.
Excellent responses, and please, students, if you don't mind, put your hands up more often.
Oxford Nanopore Technologies has revolutionized DNA sequencing by using nanopore technology to directly read DNA and RNA molecules without relying on traditional methods like strand synthesis or fluorescent imaging. At the heart of this process are protein nanopores, tiny holes embedded in a synthetic membrane which act as gateways for single-stranded DNA molecules to pass through. These nanopores are made from naturally occurring proteins like alpha-hemolysin or MSPA, which are engineered to maintain stability and precision during sequencing. The process begins with the preparation of DNA or RNA molecules, which are first fragmented and then modified with adapter molecules. This adapter includes a motor protein that plays an important role in controlling the movement of the DNA strand through the nanopore. Once loaded into the device, an electric potential is applied across the membrane, driving the negatively charged DNA through the nanopore, one nucleotide at a time. As each nucleotide, A, T, C, or G, moves through the nanopore, it temporarily disrupts the ionic current running through the pore. These disruptions are recorded as electric signals, and with each nucleotide causing a unique change in the current. The software decodes these signals and determines the sequence of RNA or DNA.
Thank you everyone for being very attentive in class. But as future bioengineers, I want to ask an open-ended question. So when we talk about nanopore technology, the technology used in ONT, considering what you already know, what would you think, how should one go about designing the nanopore to sequence DNA? Yusuf?
I'm not exactly sure, but I think I would make sure that the protein I use for sequencing the DNA is high in specificity and resolution. So what I mean is that I'd make sure that the electrical signal output is very clear and well defined, I think.
Love your answer. And you can actually see this being the case with alpha hemolysin, which has consistent pore sizes. Its structure is facilitated by protein bonding, and it has a modifiable and adjustable degree of sensitivity, which can be used for obviously different applications like DNA sequencing. In the case of MSPA, its smaller pore size provides great resolution for identifying bases and is highly resistant to environmental changes and fluctuations.
Does anyone have any other questions?
Yeah, Mister, can we genetically engineer these pores?
Absolutely can. So as I mentioned, some of these pores are modifiable using genetic engineering, where they can be altered to optimize their pore size, stability, and ability to interact with DNA and RNA. For example, motor proteins or adapters can be added to slow the DNA movement, which enhances signal clarity.
Anyone has any more questions?
Actually, I actually have a question. How are these nanopores fabricated and inserted into the flow cells?
That's an excellent question. Let's dive deep into how these nanopores are actually made and used in these sequencing devices.
Nanopores are critical components in DNA sequencing technology. They allow for the passage of ions and, when engineered appropriately, can facilitate the sequencing of DNA with high precision. We will cover the engineering and production of nanopores, their role in lipid membranes, and the function of flow cells. These nanopores, such as alpha hemolysin and MSPA, are derived from bacteria. These proteins form natural pores that permit ion passage, which is essential for DNA sequencing. The genes encoding these nanopores are inserted into host cells like bacteria or yeast. This process allows for large-scale production of nanopores through recombinant DNA technology. Once purified, these nanopores are integrated into synthetic lipid membranes. These membranes provide a stable platform for the nanopores, mimicking their natural environment in cell membranes. Lipid membranes are crucial for providing stability to the nanopores during the sequencing process. And these membranes also facilitate the flow of electric current through the nanopores, which is essential for detecting DNA. And by stabilizing the nanopores, lipid membranes ensure that the DNA sequencing system operates effectively. The flow cell is also a small chip designed to hold and distribute the sample solution, and a proper design of that cell is crucial to facilitate the interaction between DNA and nanopores. It ensures that DNA molecules are brought into contact with the embedded nanopores for effective sequencing. When DNA threads through the nanopore, it causes ionic current changes, which are critical for sequencing. The sequencing softwares play a vital role in detecting these current changes and analyzing the detector to detect accurate DNA sequencing. In summary, these nanopores play a pivotal role in DNA sequencing by allowing the detection of ionic current changes as DNA passes through them. The engineering of these nanopores, their integration, and the use of flow cells are crucial components that enhance the accuracy and efficiency of the sequencing process.
This allows the sequencing software to detect the ionic current changes caused by DNA as it threads through these nanopores. Does anyone have any questions?
Yes, Professor. Are synthetic nanopores used in ONT devices?
That's actually a great question.
Is it?
So, it's a fascinating area of research currently. But as of now, ONT primarily uses biological nanopores because of their precision and ability to interact with DNA. However, synthetic nanopores, like those made with graphene and silicon nitride, are being explored in academic settings. So these synthetic alternatives are more durable, and they're also can be customized for specific applications. But they currently lack the molecular level accuracy of biological nanopores.
Thank you so much, Professor.
Of course.
Thank you.
Yes. Professor, could you combine nanopore sequencing with other sequencing techniques?
That's a great question. We have developed something called hybrid nanopores, which are an innovative combination of biological and solid-state nanopores. In this design, a biological protein nanopore is embedded into a solid-state nanopore, which creates a hybrid system that merges the specificity and sensitivity of biological pores with the durability and stability of the solid-state nanopores.
Sounds like the best of both worlds.
It indeed is.
Anyone have any more questions?
Well, let's take a small tangent. What do you call the art of turning paper figures into something beautiful and artistic in a unique shape? What is that called? Anyone know?
Origami.
Exactly. It's called origami, and you actually see applications of origami in nanotechnology.
Go ahead.
Professor, what kind of applications does DNA origami nanopore technology have?
Hamid, I love your questions. So that's a great question. So DNA origami nanopores are especially promising for studying what we call the epigenetic markers, such as DNA methylation, or for analyzing very specific molecular interactions. So for example, because they're fully customizable, they could even be used for sequencing unusual or modified nucleotides that aren't very well detected with traditional other technologies.
Thank you, Professor.
Of course. So thank you all for your attention today. It was a very engaging, and you are all very promising students. And thank you for asking these amazing questions.
The world of nanobiotechnology and nanopores is everlasting and infinite in nature. I can't wait to see all of you young bioengineers grow up to be more senior bioengineers and to pioneer the research for future generations. Thank you all for your attention. Does anyone have any further questions? Thank you. Do you have any questions?
How do we reach out to you for networking opportunities?
I'll give you my email after class. Thank you.
Bye bye.
Thank you, professors.